Welcome back for another lecture in the human species. This lecture is focused on species and speciation and something called cladistics that I'll explain in this lecture. Okay, before we get started, I just want to say I know that taking an online course like this is kind of weird. It's kind of difficult for you, and there's some difficulties for me as well because we don't have the same kind of interaction. And so I know that things are hectic and busy, and there's some difficulty involved here. But I really do want to encourage everybody to study for the first exam on Tuesday, June 9th. The reason is that obviously everybody wants to get the highest grade possible, but the higher your grade, the easier it is for you to on the exam. The easier it will be for you to get the grade that you want at the end of the course. The less stress you'll have going through the course and completing the subsequent exams. And so what does it mean to study? I find in my experience in teaching courses like this that sometimes people sort of like pick up bad habits. And in my opinion, the best way, the thing that works for me, I think everybody has to find the thing that works for them, but the thing that works for me is writing things out by hand. So like watching these videos, reading the lecture, reading the assigned readings, and taking notes on paper with pen or pencil. And I know that that's like boring and inconvenient, but the inconvenience of that is what helps you remember it. The slow time that it takes for you to write something down is the thing that helps you remember it. There's something about the connection between like your memory and the physical act of doing something like that that's really beneficial. So in my view, what you should do is, again, watch the lecture videos, take notes, read the assigned readings, take notes, and then using the key terms and concepts list that I'm going to post on Blackboard, make sure you understand everything on the list by writing down the definitions of it and reorganizing your notes. Basically rewriting your notes from the lectures and readings in one sitting or a couple of sittings so that you can sort of put all of the information and all the material together in your mind. And then once you do that, if you want to be really sure, you can go to like a Dwayne Reed or a CVS or something like that and get a pack of index cards, 3x5 index cards to make your own flashcards. Well, why can't you just use Quizlet? Why can't you just use like an online flashcard thing? Well, it's the same reason that I just mentioned, that if you just type some definition onto a website to make a flashcard, you're not going to remember it. If you instead rewrite your notes on the flashcards, which means by that point you will have written those notes 3 times basically, right? So write the terms for all the things that we've gone over so far on those flashcards with the term on one side and the definition on the other, and go through all those flashcards. You might have 100 of them, you might have 150, you might have more than that. Go through those flashcards and start with the definition side and see if you can remember what the definition of that remember the term of that definition. Then repeat that process starting with the term side and see if you can recite the definition. If you get one wrong, set that card to the side and keep going through your stack of cards. Then go through the stack of cards of the ones that you got wrong and repeat that process until you don't get any of them wrong. And so at that point, you've memorized everything. Now, admittedly, that's like a brute force method, but it's a method that will work and will help you get a high score on this exam and on exams for any course that's kind of like this, that's not like a technical course. We're not doing like math problems and that kind of thing, right? And then if there's anything from the lecture videos or the readings that you just don't understand at all, you're like, why the hell are we talking about mitochondrial DNA or whatever it might be? Then you have, please send me an email to let me know. Otherwise, I don't have that information. I can't help you. So if there's something that you just cannot understand, you can understand why we're talking about it, what the connection to other things is, feel free to let me know. It's not the kind of thing where, hey, I didn't do the lecture thing, I didn't do the reading, can you just tell me what this is for the exam? Well, in that case, of course not. I'm not going to, that's, you have to do your part as a student as well, which I'm sure most of you are doing. Okay. I know the exam's going to go well. I know you're going to do great. You're going to study over the weekend as much as possible and get the best score that you can. Okay. So at this point, in the couple of lectures we've had so far, there are a few sort of like major topics that I think we've come to understand. The first one is the basic evolutionary process, right? Like, what is the process of alleles or gene frequencies changing within populations over time? What is the process of phenotypes changing within populations over time? And we refer to those as the forces of evolution. And of those forces, there are 4 main forces of evolution. So what are they? Take a second and try to see if you can remember what the 4 forces of evolution are. Don't look at notes. Don't look up anything. Just see if you can remember it. This isn't a test. It's just a curiosity thing. Okay, mutation is one. Genetic drift is another. Gene flow is a third. And selection is a fourth. And among selection, there are a few different ways it can happen. We have natural selection. That's like the garden variety form of selection that we usually think about. We have sexual selection, in which traits evolve that enable individuals within species to increase their fitness. Within a sex, within a species, increase their fitness, right? So in mammals, usually this is the evolution of traits among males that enable them to compete with other males, and traits that allow female individuals to choose the best male individuals. Okay, so that's the basic evolutionary process from generation to generation, how things change, how phenotypes and alleles change. We understand the basics of how traits are passed on to offspring. We know that it's through so-called particulate inheritance. People like Gregor Mendel, didn't know what genes were, didn't know what DNA was, but he knew that there were factors passed on from parent to offspring. And we today know those factors to be genetics, to be DNA. And so those are the particles, right? Particulate refers to like a so-called particle. And the particles are DNA, right? So particulate inheritance. You pass on your DNA to your kids. We understand that variation is very important for the evolutionary process, in particular natural selection. And we understand a little bit about how it's introduced into populations, in particular through the evolutionary process of mutation and through forms of what's called genetic recombination. Genetic recombination during the production of sex cells or gametes through meiosis. And so crossing over of chromosomes and random assortment of chromosomes during the process of meiosis are forms of genetic recombination. And it's why gametes have sort of unique combinations of DNA of the parent. And it's part of why it's why offspring don't look like identical sort of blends of their parents, in part. However, there are some very important topics we don't yet understand that are related to these earlier things. And the first one is, what is a species? After all, this course is named the human species. So what the hell is a species? How do new species evolve? Right? How do we understand um, evolution on such large scales of species when the evolutionary process occurs on a much smaller scale? And finally, how do we figure out how different species are related to one another? Um, you may have heard me reference the fact that the closest living relative of humans is the chimpanzee and bonobo. So why do we know what what led us to that conclusion? This is the case for both living species, like humans and chimpanzees, but it's also the case for um, extinct species that are no longer living. So those species are represented by the fossil record. So, for example, how do we know that um, a particular fossil is a human relative rather than just some other primate relative? Well, hopefully in this lecture we'll answer these three questions. Okay, so first let's talk about the scales of evolution. What do we mean by the scale of evolution? Well, we really just mean, does it happen on a small scale or a big scale? Is it happening on the level of like generation to generation? Or are we talking about hundreds of thousands or millions or tens or hundreds of millions of years? So microevolution, micro as in like small, refers to short-term changes across a few generations. And so this is the kind of evolutionary change that we can observe in our lifetime, right? We can observe different organisms and the way that the evolutionary process modifies their alleles or their phenotype, right? We can study this in a lab environment in universities and research centers. We can study this in the field, meaning in natural populations around the world. And so we spoke a little bit about, in last lecture, the evolution of larger beaks in some of the finches in response to a major drought in 1976. Right, the major drought reduced the availability of small seeds, and so individuals that just naturally, as part of normal population variation, had larger beaks, those individuals were better able to eat the available large seeds, and they survived. And because they survived, they had kids who inherited their genes that coded for the large beak size. And so therefore, over a few generations, the average beak size increased. And that's through a form of directional natural selection. And so that's what we mean by microevolution. We can observe that in just a few generations. We can document that in our lifetime. Another example of microevolution is the evolution of bacteria. We can observe basically the evolution of antibiotic resistant bacteria. And so this is why the idea of should a doctor prescribe antibiotics? Is there an over prescribing of antibiotics? And the answer is probably yes. There's probably an over prescribing of it. And so in response, the bacteria that are typically killed by antibiotics are evolving resistance to antibiotics. And that's because the ones who naturally, as part of the bacteria population, have variants that enable them to survive the antibiotic are the ones that have more bacteria kids or offspring or whatever. And so now those kids inherit that ability, that resistance to the antibiotics. And so that's microevolution. What about macroevolution? Macro refers to, it's like the opposite of micro. It's like looking at it at a larger scale. And macroevolution refers to long-term changes in features of species and life on Earth. And so in macroevolution, we're thinking about the origin and extinction of species, right? This is like large scale evolutionary change. This is the stuff of evolution. Macroevolution is typically the scale of evolutionary change that is probably most misunderstood. And that's because macroevolution is basically just an extension of microevolution. And so you can imagine, if we're thinking about the origin of of a completely new species, even if if it's true, which it is, we couldn't observe it in our lifetime. Right? Microevolution is just a few generations. It's just a short evolutionary change. But macroevolution takes thousands of years. Right? It takes hundreds of thousands of years, millions of years. We hope to live to 100 years old, or maybe more if we're extremely lucky, or not, depending on your perspective. But macroevolution has to be studied through paleontology. What is paleontology? Paleo just means old. And so paleontology is the study of old stuff, the study of old life, basically. And so we study old life that's no longer living through fossils, through basically preserved remnants of a once living organism. We can also study macroevolution through comparisons of genetics and morphology, like what an organism looks like among living species. Remember, if we have similarities and differences among living things, those similarities and differences can only be explained through the evolutionary process. Species are not different for any other reason than the evolutionary process. And so if we compare species to one another and their similarities and differences, in an indirect way, we're looking at the result of macroevolution. And so the indirect nature of the study of these longer-term evolutionary changes is more difficult for people sometimes to wrap their heads around. But as I said, macroevolution is no different from microevolution on the level of the evolutionary process. Right? Macroevolution is just adding up all of the changes that occur through microevolution over periods of thousands or hundreds of thousands or millions or more years. And that's it, right? So at the root of both micro and macroevolution is the same thing, and that's the evolutionary process, right? Drift, gene flow, mutation, and selection. Okay, so how do we identify a species, both a living species and an extinct one? Okay, so what we have is a series of species concepts. Species concepts are ideas. They lay out a set of criteria for how we define what a species is. There are many species concepts, and there are too many to focus on in this course. And so we're just going to talk about two of them. The first one is the biological species concept, also known as the BSC. It's the abbreviation for biological species concept. And so under the biological species concept, species are groups of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. So in other words, a species is a group of actually or potentially interbreeding populations that differ in terms of their reproductive isolation in some form. And so, again, that is the major criterion. That's the major defining feature of the BSC, or the biological species concept. If essentially, if two individuals can interbreed and produce fertile, healthy offspring, right? Offspring that can have their own offspring and so on, then the original mating parents are members of one biological species. If, on the other hand, the two parents interbreed and they can produce fertile offspring, but that offspring itself cannot have its own offspring, then they are not of the same biological species. So, like a horse and a donkey, for example. Actually, I'll use other examples later in this lecture, so I'll hold off on that for now. The problem with the biological species concept is that there are tons of living things outside of the animal kingdom, like bacteria, like plants, that it doesn't apply to. In some cases, you even have asexuality, where it doesn't require another individual to reproduce. It's also an issue for hybrid animals and something called ring species. I'll talk about ring species in a little while, but essentially the problem here that we encounter is that you can think of biological variation in some sense. You can think of it in a continuous way, that there's a spectrum of variation. And so a species concept and a species itself is a categorical system, right? So in other words, the nature that we are trying to categorize is actually not naturally categorical. It's actually continuous. There's a spectrum from one end to the other. And so you can, I kind of like the analogy of like the color spectrum, right? You can think of a rainbow. Don't think of a rainbow in kindergarten, right? Think of a rainbow in a college physics course, right? There's an entire spectrum of color from one end to the other. And so, even though we have red, orange, yellow, green, blue, violet as part of the rainbow, there are gradations in between. And so the species concepts are like the colors red, orange, yellow, green, blue, violet, and the nature, the animals, the plants that we're trying to describe are like those gradations between the colors, right? So each species concept is not perfectly applicable to all living things for that reason, right? We're trying to categorize what is naturally continuous. It's also, the biological species concept is also a problem for the fossil record, because we can't observe whether or not two fossil individuals could have reproduced and produced healthy and fertile offspring or not. So we can never really use the biological species concept. And again, if we want to understand evolution on a macro scale, we have to incorporate fossil data, right? We have to incorporate paleontological data. And so the biological species concept is not that useful for fossil species. Now, we could make an inference, we could make a guess, an educated guess about whether two fossil species could reproduce or not based on rough proxies or rough correlates of differences between living species that can and cannot reproduce. But that's still imperfect and not really a solution. So we have a second alternative species concept called the phylogenetic species concept, right? Yet another term, phylogenetic. Phylogenetic or phylogeny just refers to evolution. It refers to the evolutionary relationships among a group of species or a group of organisms. And so we can call this the PSC, the phylogenetic species concept. And basically, under the phylogenetic species concept, basically you're looking for things that distinguish clusters of individual organisms that can be linked to a pattern of ancestry and descent. And so, really, there are two criteria: distinguishability and descent from a single ancestral species. So, what do we actually mean by that stuff? Okay, so if we have a collection of fossils that we think are human relatives, we can ask, are there certain traits that some of them have that others don't? Are there certain traits that some individuals consistently show and others consistently lack? That is referring to this criterion, right? Are there things that allow us to distinguish one group of fossils from another? Okay. If there is such a trait or such collection of traits, do we think that collection of fossil individuals evolved from a single ancestral species? In other words, are all of those individuals with this other distinguishable trait a part of the same group? Are they a part of the same thing? If so, perhaps they are a separate, distinct biological species. Right? It wouldn't make sense to include multiple fossil individuals in one species if each of those individuals actually came from a different ancestral species. Right? In that case, we're just saying they share something, but it's not because of evolution. It's not because of evolution from a common ancestor, I should say. And so the phylogenetic species concept, it sort of relies on this idea that similarity and difference can only be explained in biology in terms of evolution. So if a group of fossil individuals are similar, and if that similarity is shared from a common ancestor, then they are likely to be a separate phylogenetic species. And so we use this when we can't use the biological species concept, which is fairly frequently in terms of fossils. We'll talk a little bit about the human fossil record later on in the course, and essentially we'll be applying the phylogenetic species concept in that case. The PSC, the phylogenetic species concept, is closely related to other species that are used in morphological and paleontological studies. And so that includes paleospecies, chronospecies, and morphospecies. Right? So again, paleo just means old. You can think of it that way. Chrono is like chronology or time. So one species evolving into another in the fossil record, we may refer to those two populations as different species, even though biologically they are continuous. Even though, the only reason that they're referred to as something different is because they're found at different times. And so, one species is the direct sort of gradation into the other. We would consider them chronospecies, maybe. Morphospecies, morph is like shape or morphology. So a morphospecies is just recognizing that one group of fossils has a unique or distinct shape compared to another one. Again, it's getting at the same criterion of distinguishability. So basically what we're saying is that we can't use the biological species concept for the fossil record. And so what we do as an alternative is simply just say, hey, are these species consistently, or are these fossils consistently different from these other fossils in a regular, consistent manner? If they are, we can assign a name to each of the two groups to say, hey, this is one species, this is another species. The actual biology of that is less clear. And so there's a really cool study that actually I just saw yesterday that looks at whether species, whether populations reproduce and let me start over. Whether populations mate and produce no offspring, whether they mate and produce infertile offspring, whether they mate and reproduce an offspring in which one of the sexes is infertile, or whether they mate and produce fertile offspring. And so in each of those categories, they looked at like genetic distance between species. And so it's no surprise that the more genetically distantly related species are, the less likely it is that they'll produce fertile offspring. And so they applied this approach to ancient DNA that's sequenced from fossil individuals that we can't observe directly. And these are fossil hominins, Neanderthals, and other archaic human relatives. And they found that based on the differences between anatomically modern human DNA and between Neanderthals, that they fell within the fertile offspring category. And that makes sense because we know that some modern human populations living today has a small percentage of their genome inherited from archaic hominins like Neanderthals. And so we know they interbred, and yet we still refer to the Neanderthal fossils and the anatomically modern human fossils as separate phylogenetic species. And that separate name that we give them recognizes the fact that Neanderthals lived in a different place, had their own way of life, and their own evolutionary history for hundreds of thousands of years that is distinct from that of anatomically modern humans. And so the name that we assign to a Neanderthal and to an anatomically modern human references the fact that their differences accumulated through the evolutionary process, right? They can be distinguished on the basis of consistent criteria. So we call them something different to recognize that and to communicate about those fossils, even though biologically they are the same species. And so the question of are they a different species or not biologically actually turns out not to be that interesting or that important, right? Because they made and used different tools. They had differences in their skeleton, in their skull shape, in their ability to make art, right? So anatomically modern humans made art, cave art, and figurines and all kinds of things, whereas Neanderthals don't seem to have done that, at least at high frequency, right? So like whether they are a true biologically distinct species actually is not that important. They didn't interact with one another except in some cases where they met and interbred anyway. But so I hope that illustrates what the phylogenetic species concept is as an alternative to the biological species concept in the fossil record. Okay, so how are new species formed? You know, how do populations become differentiated? Okay, so we referred to this process as speciation. Speciation, or the formation of new species. And like everything, you know, like the evolutionary process itself, right? This is just an extension of the evolutionary process. It doesn't occur with the snap of a finger, right? Again, this is a process that takes time. Everything that we're talking about takes time. And so the first form is called allopatric speciation. And this occurs when populations become isolated from one another geographically, meaning they live in different places due to some geographic separation. or in many cases, some natural separation, like some feature, some part of the landscape causes their separation, right? A canyon, a mountain range, a river. And so essentially what happens is, and this is a classic kind of example of chimpanzees and bonobos. But what happens is you have natural features that isolate and separate populations. And so this is the central chimpanzee, Pan troglodytes, troglodytes, right? So this is the genus, Pan, the species, troglodytes, and the subspecies, troglodytes also. The central chimpanzee is geographically isolated. It lives in a different part of Central Africa compared to bonobos and another subspecies of chimpanzee, due to the formation of a river that separates their population. The eastern chimpanzee, on the eastern part of Central Africa, is Pan troglodytes schweinfurthii, which is an interesting name. It too is separated from another population by the Congo River, and that population is the bonobo, Pan paniscus, a different species in the genus Pan. And so essentially, think about this. What happens is, because these populations are living in different places, through the evolutionary process, through mutation, genetic drift, and natural selection, each of these populations is evolving over time, right? They're accumulating their own genetic differences through mutation. Natural selection, due to differences in different environments, may favor some alleles over others. The separation of these different populations may have resulted in a loss of genetic information, or may have resulted in a loss of alleles through genetic drift. And so the point is, with geographic isolation, in this case by rivers, stops gene flow between populations. Right? Eastern chimpanzees are not mating with bonobos in the wild. And so that cessation, that stopping of gene flow, when combined with the other forces of evolution, mutation, drift, and selection, result in each of these populations over thousands of years or hundreds of thousands of years to accumulate differences. They become a little bit different from one another. And so with enough time, they can become so different that biologically they can't reproduce and produce fertile offspring. That would be a biological species concept difference. Or they accumulate some genetic and morphological differences that characterize members of each of those populations that enable us to distinguish them from one another. Right? So like bonobos are slightly smaller in body size. Especially male bonobos are slightly smaller in body size than male chimpanzees. Bonobos have different like soft tissue characteristics in terms of the hair on their heads, certain features on their heads. They look, very, very similar to chimpanzees, but they aren't the same. And those differences, again, any difference that we observe in animals, for the most part, is the result of the evolutionary process. It didn't just appear spontaneously, right? So the fact that bonobos are a little bit different from chimpanzees can only be explained by the evolutionary process. So that's allopatric speciation. Another form of speciation is parapatric speciation, another term, right? This is why it's important to write all of these things down, as I mentioned earlier. Okay, so parapatric speciation occurs in continuous populations. Continuous. And again, continuous just means that the range and the variation of the population extends continuously from one point to another, from one location to the next. And so an example of this, a really cool example, is something called ring species. Ring species. And so this is an example from salamanders. And this is the state of California, right? You got Sacramento here, San Diego down here, and so on. Essentially, the way it works is throughout the range, the continuous range of these salamanders, they start to look a little bit different, right? They're accumulating differences throughout this range. And each of these populations that neighbor one another can reproduce and produce fertile offspring, right? They're the same biological species. But by the time you get to the end of their range at this location, this population can no longer reproduce and produce fertile offspring with this population. And so through this ring of their distribution, they've accumulated differences, right? The evolutionary process is producing differences in these populations throughout this range. And by the time you get back to the starting point, so much time has passed and so many changes have occurred that these two are not the same biological species, but these two are. And so this speaks to the earlier point that I made, that a species concept, a species name itself is a category, but variation in nature often is distributed continuously. And so this kind of speciation where you've got changes accumulating throughout a continuous range, that's parapatric speciation. Okay, the final form of speciation is sympatric speciation. Sympatric speciation. Sympatric speciation occurs in the same geographic area, right? Sympatric, I remember sympatric as same geographic area. Admittedly, this is more common in plants than in animals, and it occurs through a form of selection, a mode of selection called disruptive selection that we talked about last time. So remember, if you have some trait value that's a continuous trait, we can call this trait, and we can call this like number of individuals here. If you have some normally distributed continuous trait where this is like large measures of the trait on the right hand of the x-axis, this is short measures of the trait on the left hand of the x-axis, and most individuals fall somewhere right here. This is like, in a hypothetical scenario, the mean and the median of the population, a kind of average for the population. If for some reason in the same geographic area, individuals who naturally fall at this end of the population and simultaneously individuals of this end of the population have increased evolutionary fitness, they're going to pass on those trait values. They're going to survive and pass on those trait values to their offspring, which over time results in a bimodal distribution, where you have now more individuals at the extremes of this continuous range of this trait value, and many fewer individuals in the middle. And so through time, through this process of disruptive selection, you now have populations that are accumulating differences and forming new species. And so this is an example here of the evolution of a bimodal distribution in fish, in lip size, in body shape, and the shape of the jaw, and the diet in these two forms in the same geographic area. Right, so this is not a terribly common thing, but it is something that does happen. And so here's a kind of summary of these three forms. allopatric speciation. This is geographic isolation, as in like chimpanzee and bonobo populations. Parapatric speciation, where you've got like a continuous a speciation throughout a continuous range, and sympatric speciation, same geographic area. And the thing I want to drive home is that no matter what form of speciation we're talking about, at the end of the day, all we're talking about is organismal variation. We're just talking about organisms as part of populations that have similarities and differences. And the only way we can explain those similarities and differences is through the evolutionary process, right? Similarities and differences aren't spontaneously created, they accumulate over time through a process. And so at the core of speciation is the evolutionary process. Each form of speciation occurs because of the evolutionary process. Okay, that's the last time I'll say it. Okay, so we mentioned that in the biological species concept, one of the central criteria is that populations are reproductively isolated in some way. And so the question is, how does reproductive isolation occur? And what is that? Okay, so reproductive isolating mechanisms refer to any factor that prevents two individuals from hybridizing, from two individuals from producing an offspring. That can be behavior, it could be anatomical, it could be ecological, like the environment and the interaction of the animal with the environment doesn't enable this to happen. And so we can divide these isolating mechanisms into pre-zygotic isolation and post-zygotic isolation. Remember that a zygote, hence zygotic, is a fertilized egg. So these are the things, the pre-zygotic isolating mechanisms are the factors that prevent the egg from being fertilized. Barriers to fertilization. And so that includes mate recognition, which means that one individual doesn't even realize that another individual is a potential mate, just because they're so distinct from one another. And mechanical issues, meaning that it's just mechanically, physically impossible to happen, right? We use the domestic dog is a really interesting example because dogs are all the same biological species. They look different. We have different breeds, right? We have a Chihuahua, a Great Dane, a Lab, a Dachshund, whatever, but they're all the same biological species. And so, in theory, any two dog breeds can produce fertile offspring. They're the same species, same biological species. But it's not difficult to imagine that a Great Dane and a Chihuahua probably can't produce offspring due to mechanical issues. Right? The size difference between those two breeds is too dramatic for it to happen. That's an example of a barrier to fertilization. Okay. We also have post-zygotic isolation. So post-zygotic means after the fertilization of the egg. And this includes sterility and offspring non-viability. So sterility just meaning that basically that the offspring cannot have offspring of its own. Excuse me. And so let's say two parents could mate and produce a kid, right? They could produce an offspring, but that offspring may not be able to have its own offspring. That offspring is sterile. in which case the two parents are part of different biological species. Sometimes you have a scenario where a hybrid offspring is produced, hybrid of the two parents. Hybrid just meaning like a combination of the two parents, and only one of the sexes is sterile. So this is referred to as Haldane's rule, where the heterogametic sex is sterile. What the hell is heterogametic? um, hetero just means different. And so the sex with different gametes, or different, we can think of these as different sex chromosomes, is sterile. So in mammals, males are the heterogametic sex, because males have the sex chromosomes XY. Right? So in mammals, males have XY, and females have two X chromosomes. And these, are, of course, are the sex chromosomes, which are the 23rd pair of chromosomes. So under Haldane's rule, males, in some cases, will be the sterile sex. Male individuals will not be able to have their own offspring. We can do it like this. But female individuals will be able to have their own offspring. So this is kind of like an intermediate sterility. And so one interesting example is the hybridization of lions and tigers. If you've ever seen Napoleon Dynamite, you may have heard of a liger before, but a liger is a male lion. A liger is produced when a male lion and a female tiger hybridizes, and a tigon is produced when a male tiger and a female lion reproduce and hybridize. And so you might wonder, like, if we have ligers and tigons, then why don't we have entire species that are made up of ligers and tigons? Well, the reason is Haldane's rule, that the heterogametic sex is sterile. So in other words, like, male ligers and male tigons can't have their own liger and tigon babies. And so that means that female ligers and female tigons subsequently mate and reproduce with individuals of one of the parent species. So in other words, lions and tigers are not becoming differentiated. They're not forming new species through hybridization because the heterogametic sex is sterile, even though they can produce offspring. You also have the case where the offspring is non-viable, where they can't have offspring at all. And so even though, like, two animals can of different species can mate and attempt to reproduce, if they produce an offspring, it might be completely sterile, or the male individuals are sterile, or the offspring is non-viable. So these are the mechanisms of reproductive isolation that enable us to have the biological species concept, right? Prezygotic and postzygotic isolating mechanisms. Under prezygotic, we can also think about temporal issues. Temporal just refers to time, temporal. So over time, species change. They accumulate differences through the evolutionary process, and that biological change prevents prezygotic. It acts as a prezygotic isolating mechanism. One species can't go back in time and reproduce with a previous population that no longer exists. Okay, so how do we reconstruct how species are related to one another? Well, before we get there, we have to talk about these two terms. In fact, the question of what a species is related, is kind of a branch of taxonomy. And so we have these two related concepts in biology. And those two concepts are phylogeny. We saw this word in the phylogenetic species concept, and taxonomy. So phylogeny refers to the evolutionary relationships among species. So if you have heard of the tree of life, or if you've heard me say that chimpanzees are the closest relative to humans, we're talking about phylogeny. We're talking about the branching relationships, the branching pattern of relationships among species through time and space. Phylogeny can be represented by an image that looks kind of like a tree that represents a particular educated guess about how those species are connected. You can think of like putting together a family tree for yourself and your relatives. Your family tree contains parents and daughters. Phylogenies contain parent species and daughter species. It's a similar kind of thing. Taxonomy, on the other hand, refers to the names that we give those biological entities. It's the names that we use to communicate about the tree of life. We have rules that dictate how we come up with those names. And basically, like the system that we have for assigning names to things, to living things, has to relate to how they're related to one another, has to correspond to their relationships. And so I'll talk about that in a moment. And so usually under taxonomy, we have groups of living things as species, as families, as order, and so on. And these are referred to as taxa. A single one of these is a taxon, right? So a species is a taxon. A family, not like our individual families as people, but like the family in terms of life forms or orders, like the primate order, each one of those is a taxon. And finally, those taxa should be what's called monophyletic. They should make up a monophyletic clade. That simply means that all of the taxa that we assign to a family or an order all evolved from a single common ancestor. You can think of mono as one. Like mono is the opposite to stereo. Stereo is like a left and right speaker, left and right headphones. Mono is a single speaker or a single audio source. So taxa should evolve from a single ancestor if we put them in the same group. And that makes sense, because why should we call bears, humans, and rats a unique name if they aren't each other's closest relatives? Right? If we're also excluding raccoons, lions, and chimpanzees. And so I'll explain this in graphical form in more detail. Last thing I want to say is that the purpose of taxonomy is to identify monophyletic clades. Right? I'll emphasize that again. The purpose of taxonomy is to identify monophyletic clades. We also use it to communicate. That's important as well, but really this is like at the core of taxonomy. Okay, so if phylogeny refers to how living things are related to one another, and if we can depict that, if we can show that those relationships graphically with a tree, how do we read that tree? Like, what does it actually mean? How do we use that information to inform our understanding of how they're related? It looks like this, where you've got usually individual branches, right? So this is a branch, this is a branch, this is another branch, right? The branches correspond to taxa. In this case, each branch is not a single species, but it's a much larger group. Each branch is connected to another branch. And the spot where the two branches connect refers to a branching point. It refers to a hypothetical common ancestor, right? An ancestor, a living thing, in this case, in this example, a living animal that these two things share, that these two taxa share. We mean literally that each branch evolved from a common ancestor in this case. So essentially, we determine that species are more closely related to one another if they share a more recent branching point. So in this case, take a look at this and tell me in your own voice, in your own homes and other places, which two species are most closely related to mammals? Which two species are most closely related to mammals? Or which two groups, which two taxa? Well, first, it's lizards and relatives, because we share a common ancestor that's more recent than the others. And it's also amphibians. Now here's another question. Of mammals and lizards, which of those two is more closely related to amphibians? Are mammals or lizards more closely related to amphibians based on this plot? It's neither. Mammals and lizards in this case are equally closely related to amphibians. Because they each, all three of them, share a common ancestor that is represented by this branching point. The next closest related thing is a fossil that has been assigned the name Tiktaalik, which is like an early example of like land-living animals. This is an extinct fossil species. It is closely related to amphibians, mammals, and lizards, represented by this branching point. The next closely related thing is the lungfish, which is represented by this branching point, right? So this branching point, again, reflects that each of these branches, each of these taxa, descended from a common ancestral animal. The coelacanth is the next closely related animal, and its branching point, shared with all of the other taxa, is here. And we can go on and on. It's not uncommon that the tips of the branches here are more recent in time, or it's I shouldn't say it's not uncommon. The tips of the branches refer to a more recent time period. As you follow each branch back to each of these points, these are nodes, we're going further back in time, hypothetically. So in other words, this is the oldest branching point, and this is the most recent branching point. All that matters in these images, in phylogenetic trees, is the branching pattern. The orientation of the diagram does not matter. This could be rotated on its side, it could be flipped upside down, the branches could be rotated, it doesn't matter. They all show the same thing. And so with that in mind, we have 4 different phylogenetic trees where the branches are arranged a little bit differently. These are like arranged in a diagonal pattern, whereas these are arranged in this kind of like squared off pattern. It's showing the same kind of thing. So in this example, 3 of these show the exact same branching pattern, and 1 of them shows a different branching pattern. So which of the 4 is different from the others? Take a second to think about it. Which phylogeny is different? Take a shot. So let's go through it. In number 1, taxon C is most closely related to taxon D, right? They share a common ancestor. From this common ancestor, both taxa evolved. C and D are next most closely related to B in number 1. They share a more recent common ancestor. From this common ancestor, the ancestor of C and D evolved. From this ancestor, C and D evolved. And then finally, B, C, and D are most closely related to A, which is represented by this branching point. Okay, let's go to number 2. Okay, C and D are swapped, right? D is here and C is here, but okay, they're still related. They still share the same branching point, followed by B, followed by A, so these 2 share the same thing. Okay, what about number 3? Okay, C. So this is a pretty different looking plot, but C and D are most closely related, followed by B, followed by A. So hey, guess what? This is showing the same thing. What about number 4? Okay, so we see C and D here, but if we follow their branches back, they aren't each other's closest relative. In this example, B and D are each other's closest relative. And so that is a different phylogeny. Even though the order of the branches were swapped in this one, right? C and D were swapped. Even though this group was rotated in this phylogeny, it's still showing the same thing because the phylogeny only shows the branching pattern. It only shows the fact that A is the most distantly related, followed by B, C, D, followed by C, D. And it doesn't change in any of these except for this one. Okay. Taxonomy, again, refers to the names and the categories that we use to refer to living things. And so initially, the system of taxonomy was created by this guy called Linnaeus in the 1700s. And initially, he came up with 7 original hierarchies, 7 original systems or categories. And those 7 original categories are kingdom, phylum, class, order, family, genus, and species. There are many different mnemonics to remember this, but here's one: keeping precious creatures organized for grumpy scientists. And we use this word hierarchy. What does that mean in this context? Why do we call this hierarchy? Why do we refer to this as hierarchical? Well, the reason is that as you move up from the species level to the genus, to the family, to the order, and so on, it contains more and more things. So, like in the kingdom level, it contains multiple phyla. Among phyla, which is the plural for phylum, you have multiple classes. Among a class, you have multiple orders. In an order, you have multiple families. In a family, you have multiple genera. That's the plural of genus. You don't say genuses, it's genera. And in one genus, you have multiple species. So it's a hierarchy where the kingdom has the most stuff and is the least specific, and the species has the fewest stuff and is the most specific. So, for example, if you are a panther, like this thing, you are a part of the genus Pantera or Panthera. I actually don't know the pronunciation of it. I know that there's like the heavy metal band Pantera, so that's kind of hilarious. But I guess it's Panthera. But so if you are a panther, you're a member of the family Felidae. You're a felid. You're also a carnivore. You're also a mammal. You're also a chordate. You're also an animal. So when we refer to plants and animals, we're talking about, or even the animal kingdom, we're talking about the specific taxonomic kingdom called Animalia. Those are animals. Okay, so systematics then combines phylogeny, the trees and how species are related, with the taxonomy, the names that we give them. And so we could say that systematics is the study of the diversity of life and the relationships at all levels in the taxonomic hierarchy. And so again, we can go through this, another example, another taxonomy example. So the kingdom is Animalia. That's animals. The phylum is Chordata or chordates. That means that they possess a nerve cord, like a connection extension of the brain through the body. That's a nerve cord. The class is Mammalia. Those are mammals. Mammals are warm-blooded with mammary glands that are used to produce milk to feed offspring. The order is primates, which means that and all the members of primates have an opposable thumb and big toe, with a couple of exceptions, and what's called binocular vision, right, forward-facing eyes. The family, in this case, would be Hominidae, which means they have enlarged brain size, enlarged body size, adaptation to suspensory locomotion. The genus is Homo, and which means that they are bipedal with large brains and tool-making capabilities. And the species is Sapiens, which is further enlargement of a brain, a bony chin, culture, art, symbolism, music, spaceships, the internet, and so on. This is how we fit in to the natural world. We are hominids. We are members of the great ape family. We are members of the order primates or primates. We are part of the class Mammalia. We are part of the phylum Chordata, and we are animals. We have all of these things. We have large brains. We have large body size. We have opposable thumbs, forward-facing eyes. We have mammary glands um, that are used to feed babies, right, during nursing. We have a spinal cord, etc. You may be saying, hey, we don't have an opposable big toe. We'll get to that. The reason is we're bipedal. We walk on two legs. And so we've evolved a non-opposable big toe. And so whenever you've got the genus and species for any living thing, it's referred to the binomen. Bi is like two, like bicycle, two wheels. Nomen is like name, right? So two names, Homo sapiens. The genus and species are always italicized, right? They're always written at an angle. Genus is always capitalized. Species is always lowercase. And this is really important. Alert, alert. Take note, write this down. Sapiens always has an S. I am not a Homo sapien. You are not a Homo sapien. You are a Homo sapiens. It's not plural. It's not referring to that. It's not referring to a collection of individuals. It's always Homo sapiens. Okay, so a taxon, as I mentioned, is a group assigned to a particular level in this classificatory system, and more than one taxon are taxa, right? So there are many species, many taxa within the genus Homo. There's only one living today, but there were many in the past. There are multiple taxa within the family Hominidae. And so here's an example, again, of a taxonomic system that must reflect how species are related to one another. So, the clownfish, the polar bear, the chimpanzee, and the human are all animals. They all have a nerve cord, a spinal cord. That's a connection of the brain that provides innervation to the body, sensory and motor innervation. But you should know that a fish is not a mammal. Polar bears are mammals, right? They're warm-blooded. They have fur-covered bodies. They have mammary glands. They give birth to live young rather than laying eggs. Polar bears, chimps, and humans are mammals. They're all more closely related to one another than any of them is to the clownfish. But you should know that humans are part of the primate order. Humans are more closely related to other primates than any primate or any human is to a polar bear. Humans and chimpanzees are also part of the same family called Hominidae. That's the great ape family, which also includes gorillas and orangutans. But humans, modern humans, are in a different genus, Homo, and a different species, sapiens. Whereas chimpanzees are in their own genus, Pan, as in chimpanzee, and their own species, Troglodytes. And so the way that we categorize living things into this hierarchical system of taxonomy, the names that we assign to them must reflect how they are related. We don't put clownfish and humans in the same family. We don't put polar bears and chimpanzees in the same family, excluding humans, simply because chimpanzees and polar bears have hair-covered bodies. And that's because chimpanzees and humans are more closely related. So does it make sense to put polar bears and chimps in a separate family? So remember that one of the aims of taxonomy is to identify monophyletic clades. It's to identify taxa that share a single common ancestor. And so a name that we assign, a family, a genus, a species, they have to include the descendants and its immediate ancestors. And so let's say that this is humans, chimpanzees, and gorillas. This would be a monophyletic clade because it includes the common ancestor of each of these branches. We could have cases where a group contains, or a taxonomic name includes a couple of species and only some of the descendants, but not all of them. So in this example, this point here, A, represents the common ancestor of all of these branches. So this grouping would be a monophyletic clade. This grouping would be a monophyletic clade. This grouping would be a monophyletic clade. But this grouping cannot be a monophyletic clade because it's excluding the taxa that evolved from this ancestor. We can also have the situation, and I should mention that this is called paraphyletic, that this is a paraphyletic grouping because it's excluding some of the ancestors of the common ancestor of the individuals in this clade. The other case is when the group contains multiple descendant taxa from one or more ancestors, but not the ancestors themselves. So, for example, we have 3 branches inside of this box, right? We have 1, 2, and 3, and we can trace back each of their branches until each of their branches intersects. So if we trace back 2 and 3, we trace back 1, eventually you get to this point. This is the common ancestor. This is the node, the branching point that represents the common ancestor of each of those 3 branches. And so we can't assign taxa 1, 2, and 3 to a group. We can't call them something in the taxonomic hierarchy without also including everything else, right? Every other point in the phylogeny. And so this would be a monophyletic clade. This would be a monophyletic clade. This would be a monophyletic clade. And so on. We can just keep doing this forever. And so remember how we referred to taxonomy as hierarchical? This reflects that hierarchy. Right? So this would be a monophyletic clade that contains multiple other monophyletic clades that each contain multiple other monophyletic clades, and so on. And so my hope is that phylogeny, right, the trees that we're looking at, has to reflect how things are related, and the names that we assign to those things that are related have to follow this rule of monophyly. Otherwise, it doesn't reflect nature, right? If we did this, for example, let's say that this is humans, chimpanzees, and the polar bear, and we include them in some imaginary group, and we assign them a name, we say, hey, this is like a new family of animals or something. But hey, we have gorillas over here that are actually more closely related to humans and chimps, and we have like black bears or brown bears over here, let's say. And so this grouping is not biologically real because it doesn't reflect a more recent relationship in this case. And so that's why taxonomy and phylogeny have to be connected. Okay, but how do we, more importantly, reconstruct how species are related? How do we actually know that like humans and chimps are closely related, or let's say old world monkeys are closely related to hominoids, or primates are closely related to rodents, as opposed to bears or something. Okay, so when we're talking about the fossil record, like extinct species, how do we figure out that like some extinct species is closely related to a living group? Well, we use a method called cladistics, and it's a method for reconstructing phylogeny, reconstructing how species are related based on characteristics that they have in common, based on characters that they share. However, just because they share something doesn't mean it implies a close relationship. They have to be shared, what's called derived characters that are evolutionarily new or novel. Right? So again, we don't put polar bears and chimpanzees into a natural group just because they share body hair. We do put humans and chimpanzees into a group, however, because they share an important shared derived character, which is the loss of an external tail, among many, many other traits. And so we don't base this on overall similarity, because similarities can be ancient ancestral features, right? We don't put humans and chimpanzees into a group because we both have eyeballs. And so a character is just a trait. It's some property of an organism that we hope is distinct from other properties of the organism, right? So we can think about like skeletal traits that we could observe in a fossil, like the shape of the head or characteristics of the face, many different aspects of the skeleton. But they can also be molecular characters, right? Through the form of sequences of DNA. So like we know that DNA basically has a beginning and an end, and it has a certain sequence of nitrogenous bases, right? ATCG, in some unique sequence, and so on and so forth throughout the genome. And some species will share changes in DNA sequences. And that's similar, that's the same, if for all intents and purposes, the same thing as sharing a skeletal trait. So let's say that in this case, let's say like one is human, one is chimpanzee. I mean, this would be like thousands or millions, so it wouldn't just be a few, and we can use a polar bear. Let's say the polar bear retains the ancestral genetic sequence. Well, let's say that ancestral genetic sequence was ATGA, ATCG, so on and so forth, repeating for any sequence of nitrogenous bases until the end of that stretch of DNA. Well, in this case, humans and chimpanzees both share a mutation at that location in the genome. And so, I mean, in reality, in a real world scenario, it wouldn't just be one location. It would be many different locations and many different shared stretches of DNA throughout the genome, not just for humans and chimpanzees, but for all life, for all plants and animals, and bacteria. Okay, so a character state then is just a variation of that character. So in our DNA example, the character would be whatever the nitrogenous base was at a particular locus, and the character state would be which base it was. Let's say the character is the presence or absence of an external tail. The character state then would be present or absent. Humans and chimpanzees share the derived character state of having an absent external tail, whereas all other primates, all other non-hominoid primates, share the ancestral character state of having a tail being present. A derived character state is a trait that differs from the ancestral character state. This is something that appears throughout the evolutionary process that's new. If some trait appears that's new and only a few species of many share that new evolutionary trait, they probably got that evolutionary trait from their common ancestor. They are more closely related than any of them is to the other species. An ancestral character state, which is also known as a primitive character state, is one that is presented in the common ancestor of a group. So if we use our human, chimp, and polar bear example again, the presence of eyeballs doesn't tell us whether humans, chimps, or polar bears are more closely related, because that trait, that character state, is a primitive or ancestral character state. Right? The common ancestor of those three species, no matter how they're related to one another, had eyeballs. However, if we use, again, the loss of an external tail, that's only present in apes. And so it means that humans are more closely related to chimpanzees. And again, I'm just using that example, that one particular trait of tail loss, because it's a very obvious one. Okay, so again, we don't base hypotheses of relationships on overall similarity. We only look at the shared derived characters. So again, even if two species share something, we would say it's homologous, it's shared from a common ancestor. Just because they share something doesn't mean that they're closely related. Again, the North American beaver in this example shares a hair-covered body with the duck-billed platypus, but it doesn't mean they are more closely related to one another. Instead, we have to look at the derived characters, the things that differ from the ancestral state. And so this is an example of 4 taxa. I'll explain the in-group and the out-group in just a moment, but we have the North American beaver, the red kangaroo, the duck-billed platypus, and the eastern box turtle. In this example, we have 4 traits, 4 characters that we're looking at in each of these. These are the 4 characters. Long gestation. Long gestation just means that you take a long time to develop and grow the offspring prior to birth. Gestation means that you are that a developing offspring is growing inside the mother, developing inside the mother prior to giving birth to a live offspring, rather than in an egg that was laid. Hair and mammary glands are self-explanatory, and a vertebral column just means you have bones that make up part of the spine. So all of these species have at least one of these characteristics in common, and that's having a vertebral column. So having a vertebral column is likely to be an ancestral character that all of these species inherited from a common ancestor. Remember, similarities, such as the presence of a vertebral column in all of these species, cannot be explained by any other factor than the evolutionary process. So if we assume that a vertebral column is homologous, that it was inherited from a common ancestor, we can sort of superimpose that on our phylogeny down below. So the common the ancestral node, which is here, is characterized by having a vertebral column, and then all the descendants inherit that vertebral column. Only 3 of the species have hair and mammary glands, right? Turtles don't have hair and mammary glands because they aren't mammals. And so, hey, the ones with hair and mammary glands are probably more closely related because they inherited that shared derived character from a common ancestor here. And so this character state at this node, number 2, and at all of these extant taxa differs from the primitive condition. So in other words, this is derived, right? It was derived from an ancestor and then passed on to all of its descendants. Okay, only 2 of these species have gestation, and that's because turtles and the platypus both lay eggs. They don't give birth to live young. And so we can assume then that the red kangaroo and the beaver are more closely related because they share this derived character. This is a derived character that differs from the ancestral condition of laying eggs. And then among red kangaroos, or among the remaining species, only the North American beaver has long gestation. And so, in other words, from this distribution of characters, if we can infer what is primitive for this entire group, we can reconstruct how these animals are related based on the presence of new characters, derived characters that only some of these animals share. Okay, so usually when we're doing this, we define what's called an in-group and an out-group. The in-group is the group of interest in our analysis. The out-group is like a reference group, and it's hypothesized to be closely related to the in-group, but not part of it. And the purpose of the out-group is to determine what's called character polarity. So, like, polarity is like polarized, like one side's different from the other. So character polarity just means which character states, which traits are ancestral and which are derived. So in our example here, the turtle is the outgroup because it's so different from the other species that it's almost certainly not part of whatever group this is. And so the character states that the turtle has defines what is primitive for this group. And so it has a vertebral column, yes. It does not have hair or mammary glands. It does not have gestation. It does not have long gestation. Therefore, these are all derived characters. So here's a different group. It's a similar kind of thing. We have an in-group and two candidate out-groups. So the in-group contains the mouse, the kangaroo, the robin and chickadee. These are birds. The crocodile and turtle. And then the candidate out-group are aquatic, right? Salamander, or salamander's not aquatic, but the tuna is aquatic, and the salamander is not. The character that we're looking at is whether the animal gives birth to live young, they give birth to babies that are alive, or whether they lay eggs that hatch. And so all of these taxa lay eggs except for the mouse and kangaroo, which give birth to live young. They don't lay eggs. And so the candidate out group, so the question we're trying to answer here is, did egg-laying animals evolve from ancestors that gave birth to live young? Or did animals who give birth to live young evolve from egg-laying animals? The question is, which of the character states is primitive and what's derived? So here's our in-group and out-group phylogeny. In this case, the phylogeny has already been reconstructed. Here's one option where laying eggs is the primitive condition for all the animals, and live young evolves on the branch leading to kangaroos and mice. Here's the alternative where giving birth to live young is the ancestral or primitive condition, and all the descendants inherit giving birth to live young, and kangaroos and mice just retain that ancestral condition, in which case the tuna, the salamander, and the turtle, crocodile, and birds would each independently have to evolve egg-laying. So the question is, which is more likely to have occurred? This one or this one? Well, we use something called the principle of parsimony, which means that the simplest of these two competing explanations is most likely to be correct. Another way you can think of this is Occam's razor. And so when we say simplest, in this context, we mean the fewest evolutionary changes. So in this case, this option involves the fewest evolutionary changes. It doesn't require the independent evolution of egg laying across multiple different clades in different times and places. It's much simpler if all of these species just retain the ancestral condition of laying eggs, and one of the branches evolved giving birth to live young. And this is the true answer. Mammals, including kangaroos, mice, most mammals, I should say, placental mammals, humans, right? Carnivorans, rodents, primates, whatever, all the mammals don't lay eggs, right? We experience gestation and birth of live young, whereas all the other guys lay eggs. Okay, here's kind of like a toy example of how we would reconstruct a phylogenetic relationship among taxa. This is a really simple example. It's a little bit flawed because we should have more taxa, but I don't want to include a lot of taxa because it complicates things for the purpose of our example. We would have many more characters as well. We wouldn't just have, 6 or 7, we'd have hundreds, or we'd have hundreds or thousands of DNA sequences. But in this case, we have 3 taxa. We have humans, chimpanzees, and baboons. Baboons are old world monkeys that are primates. We expect that baboons are closely related to humans and chimps, but we don't think they're part of the human and chimp group. Therefore, the baboon in our example is the outgroup. Ideally, we would, we might even choose a more distantly related animal, like a New World monkey, but this will work for now. So we can go through each of our taxa and determine what the character state is for each of the characters. So the first one's mammary glands. And humans, chimps, and baboons all have mammary glands. They're all mammals. They all feed their babies with milk. So that's something they all share. Is that a derived character, though? Does that tell you anything about relationships? No, because the baboon is the outgroup. It defines what the ancestral characteristic is. So only character states that are different from this one are derived and are meaningful in reconstructing relationships. Okay, what about the presence of 5 fingers and 5 toes? Present. Humans, chimps, and baboons have 5 fingers and 5 toes. Lots of mammals have 5 fingers and 5 toes. Doesn't tell you anything about relationships. What about the presence of an external tail? Baboons have an external tail like most primates, but humans and chimpanzees do not. And so because this is the ancestral character state, it means that this is a derived character state. What about axillary glands? These are glands in the axilla, the kind of like the armpit region. Well, baboons do not have axillary glands, but humans and chimpanzees do. Okay, what about arms longer than legs? Baboons are quadrupeds. They have arms and legs about the same length. Chimpanzees are more suspensory. They hang from trees and climb and knuckle walk, and so they have arms longer than legs. Humans, of course, have really long legs, so we don't have long arms, so that's absent. What about a hair-covered body? Like, by hair-covered, we mean completely covered. Baboons have a hair-covered body, that's primitive. Chimpanzees have a hair-covered body, that's primitive. Humans do not have a hair-covered body. That differs from this character state, therefore it's derived. And finally, what about bipedalism? Walking on 2 legs. Baboons and chimpanzees have the ability to walk on two legs if they try to do it, but they don't do it. They're not obligated to do it. Their anatomy is not specialized to do it. Only humans among primates are adapted to bipedal locomotion. So again, if we look at our baboon, our outgroup, which defines what is primitive or ancestral for this collection of taxa, humans and chimpanzees share this character state. They share this new character state that differs from what the baboon has. It's shared derived. Burn this into the brain. This is a shared derived character state which has meaning. This is a shared derived character state which has meaning. Both of these character states in this collection support a close relationship between humans and chimps. Again, it's not just these two things, it's many, 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 many characters, and that includes both morphology and molecular DNA. Okay, so with this collection of species, we have a few different options of how they might be related. And so the question is, given our distribution of characters and character states, which of these options is the most likely? Here's option A. Humans and chimpanzees and baboons are more closely related. Option B, humans and baboons are more closely related. Or option C, humans and chimpanzees are more closely related. Okay, so what if we proceed with option A? We can take the characters and character states that we just identified, and we can map them onto the tree to see how many evolutionary changes is required to produce this phylogeny. Remember that the baboon as an outgroup determines what is primitive or ancestral for these species, and that includes having mammary glands, having 5 fingers, having an external tail, and having a hair-covered body. So in this case, because humans and chimpanzees don't have they don't have external tails, and because they have axillary glands, we can add this evolutionary change, which all the descendants would inherit, to the phylogeny. Humans would evolve loss of body hair and bipedalism. Chimpanzees would evolve long arms for hanging from branches. But baboons would have to lose a tail, they'd have to re-evolve a tail through a form of an evolutionary reversal. A reversal, and we say, reversal, we mean reverting back to the ancestral character state. And they'd have to lose axillary glands, another form of a reversal. And so this is 7 evolutionary events, right? 1, 2, 3, 4, 5, 6, 7, and 2 reversals. 2 of the 7 are reversals, which are not as common in patterns of evolution. The reversals are a form of independent evolution. They're a form of homoplasy. And so in these kinds of analyses, when we're evaluating them, we tend to try to eliminate homoplasy because it's less likely than traits being passed on through homology. Okay, what about option B? Humans and baboons are more closely related. We have the primitive or ancestral traits still the same that we defined by the baboon. In this case, you have both humans and chimpanzees evolving external tail loss and axillary glands independently. Chimps evolve long arms. Humans evolve loss of body hair and bipedalism. And this is still 7 evolutionary events. And homoplasy, right? Independent evolution or convergence between chimpanzees and humans. What about this option, option C, where humans and chimpanzees are more closely related? Again, we have the same primitive or ancestral characteristics defined by our baboon outgroup. Humans and chimpanzees, the common ancestor of humans and chimpanzees, would evolve tail loss and axillary glands. Chimps would evolve long arms, and humans would evolve loss of body hair and bipedalism. And so this is 5 evolutionary events instead of 7, and there's no homoplasy. And so this is the option. I gave it away. Which of these phylogenetic hypotheses is the most parsimonious? Hypothesis 1, with chimpanzees and baboons as close relatives. Hypothesis 2, as baboons and humans as close relatives. And option 3, chimpanzees and humans as close relatives. And the most parsimonious one is hypothesis 3, because it only requires 5 evolutionary events and no independent evolution, no homoplasy. And so I do want to mention that in this hypothetical example, we're saying that what's simpler is true. Does it mean that the simplest explanation is always true? Like universally, capital T, this is the truth? No, of course not. In this case, however, the relationship between chimps and humans is based on a lot more data, a lot more characters and character states than just these, 7 things that we're talking about here. But in like examples of paleontology where fossil species are hypothesized to be related in a particular manner, the large-scale results of these kinds of studies are solid, but we can debate the smaller-scale species-level designations. So, for example, with some of the fossils, human relatives that we'll talk about, nobody on the planet Earth doubts that they are human relatives, but there is disagreement about which of them is more closely related to humans directly as opposed to more distantly. Nobody thinks that they aren't human relatives. Okay, so given this more parsimonious phylogenetic relationship between humans and chimpanzees, we can say that the monophyletic clade Hominidae, including humans and chimps, is supported by at least 2 shared derived characters. Again, we can assign chimpanzees and humans to a family called Hominidae that contains other species like gorillas and orangutans, as well as the common ancestor, including those species. We assign baboons and other monkeys in Africa and parts of Asia to a different family, which happens to be called Cercopithecidae. You don't have to remember this. But so now we have 2 genera, Homo and Pan, that are both within the same family, Hominidae, that differs from a 3rd genus, Papio of the baboon, which is in its own family called Cercopithecidae. Now, what about this? What if we got the wrong phylogeny? What if we thought that chimps and baboons were more closely related because we analyzed our data poorly or because we made poor decisions in our character states? A Pan and Papio group, this gray box, if we named them a particular, let's say we called them family Panpapio. We can just make that up. Panpapioidae. Because families usually end with a day. That would not reflect their true phylogenetic relationship. If we refer to this hypothetical, Panpapioidae family in the context of how they're really related, this would be a paraphyletic group. Right? This is an example of an incorrect taxonomy, an incorrect classification based on an incorrect phylogeny. That becomes clear when we view this in the context of the true phylogeny. This case is paraphyletic because it excludes one of the descendants of the common ancestor that Pan and Papio share. It excludes Homo sapiens. So to fix this, what we would do is we assign all of these groups to a true monophyletic group containing the common ancestor and all of the descendants, Papio, Pan, and Homo. And this is a group that actually exists. It's called Catarrhini, or Catarrhines. It's the old world monkeys, apes, and humans. Now, we're excluding lots of taxa just to make this simple. But within the group Catarrhines, we can have the different families, Cercopithecidae and Hominidae, that each contain different genera, Papio, Pan, and Homo. And so this is a reflection of the fact that the names that we assign to living things must reflect how they are related to one another, right? Taxonomy, the names, has to reflect phylogeny, the evolutionary relationships. And so we can expand that out even larger to an entire order, which is the order Primates, right? So we have the entire order Primates, which includes a group here called Strepsirrhines, a group here, a clade here called Haplorhines, a group here called Platyrrhines, the Catarrhine group that we just talked about that contains Cercopithecids, old world monkeys, hominids, great apes, and so on and so forth. Okay, so what is a species? We use the biological and phylogenetic species concepts to make that determination in different contexts. The phylogenetic species concept is important for the fossil record, and that's because we can't observe the mating behavior of extinct species. We can't use the biological species concept. New species evolve through the evolutionary process in allopatric, parapatric, and sympatric speciation. And we reconstruct how species are related to one another by relying on shared derived characters under the cladistic method. And those characters can come from morphology, can come from shape, like the shape of their bones, shape of their teeth, other aspects of external phenotype, and or sequences of DNA, the particular patterns of nitrogenous bases encoded within their DNA. Taxonomy, how we identify and name species, has to reflect how the species are related to one another. It has to reflect their phylogenetic relationships that we reconstruct using cladistics. And again, cladistics emphasize shared derived characters. And so that's it. Hope everybody's doing well. I'm gonna post a key terms and concepts document today so that everybody can study for the exam on Tuesday. I'll talk to you guys next time.